Welcome to physiology. We will talk about the urinary system. Let's see. Okay. Urinary system. So urinary system can also, well, you know that the adjective here, it can be renal, or the other one is less common. You may call it nephro. All these they all relate to kidneys. So the urinary system basically is the study of the kidney functions. We'll talk a lot about this, and then functions of kidneys. There are at least 7 functions of kidneys. First one is to regulate ECF volume and osmolarity. So here we use the word ECF because if you remember, ECF consists of your blood and also your interstitial fluid. So yes, kidney does regulate your blood volume and your interstitial fluid volume. Your kidneys also regulate water and ion. Regulate sodium, potassium, calcium, hydrogen ion, phosphate or bicarbonate, phosphate. The major one is sodium. So sodium is the salt, is the ion that is in mostly occur in the extracellular fluid, and we know that outside of the cell we have more sodium, and inside of the cell we have more potassium. So and the way kidney regulate is that it control the direction of absorption of sodium, and then indirectly control the movement of water. So this is actually the principle behind for the entire urinary system. The kidney control the where sodium go. You know, the sodium can go to kidney, or the sodium can go to the blood. so if it goes to kidney, if sodium go to kidney, then water follows by osmosis, then you have more urine. If sodium goes to the blood, then it makes the blood more concentrated, then water follows by osmosis, then you will have increase in blood volume. So it is mainly this one that we'll focus on. Removes metabolic waste. For example, amino acid, excess amino acid, AA means amino acid, it will become a urea. We learned about this, the deamination process in the metabolism. And then nucleic acid, Na stands for nucleic acid, will become a uric acid. And then creatine, become creatinine. And then remove foreign chemical. Both ingest or inhale. What it means in here is that foreign chemical can be the medication that we take. Well, most of the over-counter drug, well, they are safe, not only because the active ingredient is not that much, it have very little active ingredient, but also the our kidney can remove the over-counter drug. So that makes over-counter drug very safe. But for prescription drug, well, there are lots of reasons we need a doctor's prescription for a certain drug. For one, the active ingredient, they may be in a higher dosage. And for two, kidney may not be able to completely remove them. Number three, I would say that number three is the most important point. Many of the prescription drugs, they have a lot of side effect. Yeah. So but over-counter drug, they're safe. But even though they're safe, doesn't mean that you can take as many as you want. All right. And then secrete hormone. I hope you still remember your endocrine system lecture. Remember the hormone that the urinary system secrete? It is called erythropoietin. It is also known as EPO. What is the function of EPO? Does anyone remember? It is the red blood cell production. And then it is not really secrete, but we also put it here. Kidney has a function to activate vitamin D3. Okay, I will emphasize here, activate vitamin D. To become calcitriol. Calcitriol is the active form of vitamin D, and then it can increase calcium absorption. Yeah, as we all know that if we expose our body to sunlight, then our skin will make a vitamin D, and then we will talk about the vitamin D pathway in the urinary system, and then secrete enzyme renin. We have been seeing this term renin repeatedly, okay? For example, in the cardiovascular system, I mentioned that one of the anti-hypertension drug, it works on, it is the ACE inhibitor. ACE inhibitor, I hope you still remember. Angiotensin converting enzyme inhibitor, and renin, it is upstream to that pathway. Renin is upstream to that pathway. Basically, renin convert angiotensinogen to angiotensin 1, and then angiotensin, and then the ACE convert angiotensin 1 to angiotensin 2. So we will talk about this pathway in the urinary system. We call it RAA pathway. It is you will be tested in the exam for the RAA pathway. Number 7. I would say glycogenolysis. Yeah, kidney actually store glycogen. Well, maybe I should put it this way. Stores glycogen. And then you do the glycogenolysis. And to increase the glucose level. Okay, yeah, we always say that glycogen stored in the liver and muscle, but this is like the third location that we seldom talk about. All right, let's take a look at some a picture from the PDF file, and it's like a review of the anatomy. So let me see in here. Well, you should all know this kidney structure, okay? We all have learned this from anatomy. I'm not gonna talk too much about this one. We will be focusing on the nephron. Nephron is the part that I want you to focus on. And then this is the nephron structure, and you do need to know this very well. The reason is that when we talk about the physiology of the nephron, you need to know the name of the structure. You need to know the name of the structure here. That's why anatomy is the prerequisite for the physiology. We all start from here, okay? You remember the renal artery, renal vein. Okay, renal artery will split, and then it will become different name in the kidney, and then eventually it will become the afferent arterial. The afferent arterial will go into the capillary network. This capillary network is called glomerulus. Okay, glomerulus, it is surrounded by Bowman capsule, and then inside Bowman capsule is called Bowman space. Okay, but nowadays they don't call it Bowman capsule, Bowman space. They have a new name. Okay, they call it renal capsule, renal space, and then all together they're called renal corpuscles. Okay, and the afferent, and after that the exit will be the afferent, and then usually the afferent arterial has a larger diameter than the afferent one. And then because of that, you create a positive pressure in the capillary so that you will favor filtration, okay? Remember, we talk a lot about filtration and absorption in the cardiovascular system. And in here, we will distinguish between filtration and secretion. And then we also have absorption in here. Filtration occurs only in the renal corpuscles. For this lecture, I'll call Bowman capsule, Bowman space, and glomerulus, because this is what I learned many years ago. But nowadays, they have a new name, as I say, called renal capsules, renal space, etc. So I'll just go with the old name, but the new name, they have a new name here. So filtration occurs from the glomerulus to the Bowman space, okay? From the glomerulus to the Bowman space. And then after that, we'll talk about what will be filtered, what will left behind in the blood vessels. And then all the filtrate will go through this. And this is the proximal convoluted tubule. As you know that why is it so convoluted? Because we have a lot of capillaries surrounding here. The capillary, we call this peritubular capillary. Peritubular capillaries. And the proximal convoluted tubule, all the convolution in the proximal convoluted tubule, they will increase the surface area for exchanging the substances here. And then after that, the proximal convoluted tubule will decrease in diameter. The decreasing in diameter will cause a positive pressure again in this descending limb of loop of Henle. They call it Henle's loop. Loop of Henle is also okay. The descending limb of loop is a smaller diameter. When the filtrate go from large diameter to small diameter, it will create a positive pressure, and that will also favor. In this case, we don't call it filtration. We call it this. Let me see. Oh, okay. This is not filtration. This is not secretion. This is called absorption. The reason why we call it absorption because in here we also have some blood vessels here. And the blood vessels, they're actually continuous here. And the blood vessels, they have some vertical blood vessels here. There's some vertical blood vessels here. We call these blood vessels vasa recta. Vasa is like vasoconstriction, vasodilation. It's the same prefix, means blood vessels. Recta is like rectum. Rectum is the vertical part of your large intestine. It is the not vertical part. It is means straight. Okay, straight. Rectus femoris. Okay, rectus femoris is a straight. So these some strict blood vessels here. as I told you that the positive pressure created by this change of diameter or in the loop of Henle will favor absorption, meaning that the filtrate will go from the loop of Henle back to the blood vessels. And then the ascending limb here, and then after this you have the distal convoluted tubule. Again, the distal convoluted tubule, we will also have blood vessels surrounding it, the distal convoluted tubule, and the blood vessels surrounding the distal convoluted tubule is also called peritubular capillary. The reason peri means around it, surrounding, around, you know, perimeter, okay? It's from the same prefix, tubular, tubule means the tube. So some capillaries surrounding the tube, okay? What tube? Distal convoluted tubule or proximal convoluted tubule. And yes, the distal convoluted tubule is in direct physical contact with the afferent arteriole. We'll talk about the what we call it juxtaglomerular apparatus, okay? Juxtaglomerular apparatus, which is in here. We'll talk more about this one. It consists of a macula densa and juxtaglomerular cells. We will talk about those very soon. And then the filtrate continue to go to the connecting tubule, cortical collecting duct. Cortical connecting duct, it means that these ducts, they are in the renal cortex, okay? And then as it goes down, you have the medullary collecting duct, meaning that they are in the medulla of the kidney. And then after that, they will they'll join with other nephron, and then they will eventually go to the minor calyx, major calyx, etc. And then yes, you need to know the name of all this because we will talk about all this. And then let me see. No drawing. Renal corpuscle. Okay, let's take a closer look at the renal corpuscles. You see that the afferent arteriole, it is relatively larger than the afferent arteriole, okay? That will create a positive pressure in the glomerulus, which glomerulus is the capillary here. And you see that the capillary glomerulus is wrapped by something. We call it podocyte, okay? Pod means the feet, the foot, site means cell. So these cells, they have feet, okay? They use their feet. You see all this, they are the feet. They use their feet to wrap around the capillary. Why is that? Well, we know that capillary has an intercellular cleft, and it is for the substances to go through the capillary, to go from the capillary into the ECF, okay? But in here, by wrapping the capillary with the podocyte, it actually makes the filtration more selective. It makes the filtration more selective. When you wrap the capillary here, the amount of substances that can be filtered will be more selective. So not everything can go through this, and not everything can go through the glomerulus to go into the Bowman space here. Okay. Oh, okay. We'll talk about this. We'll talk about this just in just a moment. Let's go back to the whiteboard. Okay, let's talk about the nephron. Okay, it's just recap what we just talked about. The nephron. We have glomerulus. We have glomerulus. Glomerulus, we have afferent, arterial. They, in general, they are large in diameter, and then the afferent, the exit one, afferent, arterial, they have the small one. Because of that, it will create a high blood pressure. High blood pressure in the glomerulus, the capillary. In the glomerulus, the capillary. Let me see. Where is it? And then we will have the afferent arterial. After the afferent arterial exit the glomerulus, it will become a peritubular capillary. Peritubular capillaries. Peritubular capillaries, they do both secretion and absorption. In the glomerulus, we do filtration here. So I have to specify in here. Glomerulus is a filtration occurs only in the glomerulus, and then when in other parts of the nephron we will have secretion. Okay, so secretion occur in other parts of the nephron. Filtration occur only in the glomerulus, and then the afferent arterial will also become the vasa recta that I mentioned. Vasa means straight, recta means vasa means blood vessels, recta means straight. So they are located in the it is mainly for absorption in loop of Henle. Now we will talk about juxtaglomerular apparatus. Let's take a look at the PDF file. We already seen it. So in here we have the juxtaglomerular apparatus. Juxta means next to. Glomerular, of course, it means glomerulus. So juxtaglomerular apparatus, it means it is an apparatus right next to the glomerulus. So you can see that here the afferent arteriole is larger than the afferent. And then on the wall of the afferent arteriole, we have some special cells here. Originally, the afferent arteriole, they should have the simple squamous epithelium. The simple squamous epithelium forms the endothelial cell that is in direct contact with the blood. But some of these cells, they are actually not simple squamous epithelium. They are called juxtaglomerular cell. The juxtaglomerular cell, they actually sense the stretching of the afferent arteriole. Yes, remember afferent arteriole, they do not have the connective tissues and they have elastin, so they are very good at vasodilation and vasoconstriction. And because of that, we can vasodilate, then you increase the diameter, increase the blood flow in here. When we vasodilate here, then it will increase the filtration in the glomerulus. On the other hand, when we vasoconstrict the afferent arteriole so that the pressure differences between the afferent and the efferent, they are decreasing the pressure differences between the two, then it will decrease the filtration here. So vasodilation will increase filtration, vasoconstriction will decrease filtration. How does our body monitor that? It is by this juxtaglomerular cell. The juxtaglomerular cell can detect the stretching, meaning that whether the afferent arteriole, they are vasodilate or vasoconstrict. And then what is this? This is actually the distal convoluted tubule. Remember I told you that the distal convoluted tubule has a direct physical contact with the afferent arteriole. And then this is actually the distal convoluted tubule. Distal convoluted tubule, DCT, okay? And then the cell on the distal convoluted tubule that has a direct physical contact with the juxtaglomerular cell, we call this cell macula densa. So macula densa is on the DCT, juxtaglomerular cell is on the afferent arteriole, and they are in physical contact with each other. And what does macula densa do? They detect the sodium concentration in the incoming blood. So let's go back to the whiteboard. Juxtaglomerular apparatus, we have a macula densa. Macula densa, they are located on the DCT. Their function, they are actually chemical receptor, chemoreceptor, okay, chemoreceptor. They are chemoreceptor to detect a sodium level in the incoming blood. And then we have juxtaglomerular cell. Juxtaglomerular cells, aka granular cell. They are located in the afferent arterial arterial, and their function is a baroreceptor. The baroreceptor, of course, they detect the stretching of the afferent arterial, and they also secrete. There's a second function. Maybe I'll put 1 here, and 2 here. Second function is to secrete renin. Again, you see this enzyme renin here. We will talk about the RAA pathway, the function of renin very soon. So in the endocrine system, we said that, oh, the kidney secrete renin. Renin is an enzyme, but where? Where does it exactly secrete? It is from the afferent arterial, okay? Let's take a look at some other pictures. Okay, 6 and 7, okay. In here, you can see that, okay, the 3 major processes occurring in the nephron. Basically, it's filtration, secretion, and reabsorption, or you may call it absorption, okay? Filtration, as I mentioned, it is specific in the Bowman capsule, in the Bowman renal corpuscles. It is specifically from the glomerulus to the Bowman space, from the glomerulus to the Bowman space, from the blood to the ECF. This is what we call filtration. And then secretion is also from the blood to the ECF, but it occurs in different part of the nephron. So as long as it is the same direction from the blood to the ECF, then it is the secretion. As long as it is not in the renal corpuscles. And then number 3 is the reabsorption, or you may call it absorption. Reabsorption, well, it's the same direction from the ECF back to the blood. Okay, from the ECF back to the blood, you will see secretion and absorption in many different parts. Okay, all along the nephron, And then here, this is the general substances that can filter and also that will also reabsorb or secrete. Okay, for example, drugs or medication that we take, as I mentioned, that I'll assume this is over-counter drug. So in the glomerulus, you have filtration occur. Some of that will be secreted, but along the entire nephron, should be 100% of the over-counter drug. They will be secreted to the ECF, to the renal fluid. Yeah, renal fluid, and the future in renal fluid, the filtrate or the renal fluid, they will become urine. So this is the ideal case. If substance X is an over-counter drug, then all the drug, they will be removed from our system so that it will be safe. And then substance Y can be ion and water. Ion and water, again, in the glomerulus, you have filtration. And then in the all along the nephron, you have secretion here. Oh, sorry, not secretion. This is absorption, reabsorption or absorption here. So it really depends on your body needs, okay? If your body need more water, then what will happen? Majority of the sodium will be reabsorbed, making your blood highly concentrated. Then you will reabsorb majority of the water in the filtrate back to the blood vessels. Or if your body has, remember, the low calcium level, then we secrete a PTH, parathyroid hormone, that will cause increase in the absorption of calcium here back to the blood. And then last but not least, glucose, amino acid, those are the monomers we are called nutrient. We call them nutrient. And what happened is that in here, originally for glucose, 100% of the glucose, they will be filtered into the ECF or the renal fluid. But then 100% of the glucose will be reabsorbed back to the blood. Because if we have glucose in the urine, that means you are diabetic. Okay. Okay, so we'll go back to the PowerPoint, the whiteboard. Whiteboard, okay. So we have let's talk about glomerular filtration. This is everything start in glomerular filtration. For glomerular filtration, we need to talk about different three different components. One is called hydrostatic pressure. Hydrostatic pressure of glomerular capillaries. So we call it PGC. We talk about hydrostatic pressure in the cardiovascular system. We have the hydrostatic pressure from the capillary and hydrostatic pressure from the interstitial fluid. So hydrostatic pressure in the capillary is PC, hydrostatic pressure of the interstitial fluid is PIF. So here it is the glomerular capillary, so we call it PGC. And then PGC is around 60 millimeter mercury. And then the direction is basically from glomerular capillaries to Bowman space. Second one is the hydrostatic pressure of Bowman space. We call this PBS. It is approximately 15 millimeter mercury. And the direction is from Bowman space to glomerular capillaries. The last one, we have the osmotic pressure of glomerular capillaries. It is pi GC which is approximately 29 millimeter mercury. And then direction osmotic pressure of the glomerular capillary. Osmotic pressure, if you remember the cardiovascular system lecture, I generalize, I simplify it as to assume that this is like the concentration of the blood. So you'll be going from a Bowman's capsule Bowman space to glomerular capillaries. So if in general, you know that we have plasma protein in the blood that makes the blood concentrated. When the blood is concentrated, then it will draw water by osmosis from the ECF. Bowman space, you may assume that is the ECF. So water will go from the ECF to the blood by osmosis. And then there is, I put a parenthesis here, number 4. There, theoretically, there should be a 4th term called osmotic pressure of Bowman space. Theoretically, it should be pi bs, but the value is 0. Direction, of course, from the blood, glomerular capillaries to Bowman space. This should be a bigger parenthesis. I put a gigantic parenthesis here. The reason is that it is 0. Why is it 0? Because in the filtrate, in the renal fluid, in the Bowman space, in the nephron, all the renal fluid in the nephron, theoretically there should not be any protein. Okay, if you have protein in the renal fluid in the filtrate, that means it will be called proteinuria. That means you have proteinuria, which is not a good idea. That means you may have some damage to the nephron or blood vessel system. That's why you have the proteinuria. In a normal healthy individual, this osmotic pressure of Bowman space should be 0. And remember what we learned about filtration rate from net filtration from the cardiovascular system. It is exactly the same thing. The net filtration. The net filtration, well, in case you don't remember, let me see. Let me show you this. cardiovascular system 2. Oh, here. This is the part that I'm talking about. We learned this from the cardiovascular system. Now we are using exactly the same thing. The net filtration pressure, it is the differences in the hydrostatic pressure and minus the differences of the osmotic pressure. So it is exactly the same thing that we are talking about in the right now in the renal system, in the urinary system. Let me see. Okay, net filtration pressure. Net filtration pressure, we may call it P net, is equal to the differences of the hydrostatic pressure, which is the PGC minus PBS, and then minus the differences of the osmotic pressure, okay? Pi GC minus pi BS. So when we plug in all the number, okay, PGC is 60 minus 15 minus, what is pi GC? Pi GC is 29 minus 0. Then you will have a positive value, 16 millimeter mercury. Do you remember what positive means? Positive means that it favors filtration. Positive means it favors filtration. Oh, where is it? Hold on. So positive means filtration. Running out of space. Okay. Positive means filtration. What does filtration means? Filtration means that it goes from the blood into ECF. From blood into ECF, we call it filtration. Okay. So let me see. Right now, let's take a short break. Let's take a short break. We will come back after the break. Okay.